I am very happy to be here. I want to thank the organizers for giving me the opportunity to present my research. And earlier today, we heard some wonderful presentations on circuit mechanisms underlying mental disorders. I will be discussing a different aspect, focusing on the processes occurring within single neurons. I hope to illustrate that understanding signaling pathways is critical for us to understand brain function. So my research focuses on the striatum, which is part of the basal ganglia. This brain region is critical for motor learning, such as dancing or playing a musical instrument, skill learning, including the skill of whole cell patch clamp electrophysiology, but it's also involved in some devastating diseases, such as Parkinson's disease and addiction. Now, the striatum is a fascinating brain region because it integrates information from all of cortex, and it also receives these critical dopamine inputs from the substantia nigra pars compacta. And dopamine is absolutely critical for learning and synaptic plasticity, as we've heard earlier. So a quick question is, how does dopamine contribute to learning and synaptic plasticity in the striatum? And I'm showing you here a spiny projection neuron. It's the major neuron in the striatum, and some dopamine neurons. And these images were made by my collaborator, Dietmar Plentz. So my idea of what's going on is that sensory inputs, such as this picture of a dog, or movements and cognitive states, are encoding as patterns of cortical inputs to striatal neurons. So perhaps the eye represents it, transforms into an input to a spine here, and maybe the eyebrow to a different input. And then rewards will increase release of dopamine in the striatum. And so reward of my choice is chocolate. And this is going to be given to me, not dogs. I'm a veterinarian, so I know not to give chocolate to dogs. And a key question is, how does the conjunction of cortical and dopamine inputs cause learning and change in connection strength? So for learning, I'm going to focus on synaptic plasticity because it's one of the cellular mechanisms of memory storage. And I'm sure you've heard a lot about synaptic plasticity during this course. I'm just going to provide a brief definition, which is a use-dependent change in connection strength between neurons. And we generally talk about two types: long-term potentiation, or LTP, which is an increase in connection strength, and LTD, or long-term depression, which is a decrease in connection strength. And of course, I'm interested in plasticity of cortical striatal afferents onto these spiny projection neurons. So on the right, I'm showing you traces from an experiment. So we're recording the membrane potential in current clamp. This is a stimulation artifact, and it produces this excitatory postsynaptic potential. And if you record this without, at low frequency, the amplitude is very consistent. And so we're going to call this amplitude, this baseline amplitude, 100%. And now if you give some sort of induction protocol, which is typically a higher frequency stimulation, and then you go back to recording these, you can either see an increase in the amplitude or a decrease in the amplitude. And what you see here is that amplitude normalized to the baseline. And of course, long-term potentiation is a long-term increase, and long-term depression is a long-term decrease. Now, why is this relevant for drug and alcohol use? Well, let's just talk a little bit about ethanol, or mechanisms of relapse. So with both drugs and alcohol abuse, we know that we can sometimes get people into a program where they go through withdrawal, which happens to be very, very unpleasant and contributes to the use. But once they get over that hump, they're not out of the woods. There have been some neuroadaptations. And because of those adaptations, and part of those are in our synapses, when exposed to the cues, the environment that was formerly associated with the drug or alcohol use, or even a single trial, a single consumption of the drug or ethanol, produces relapse. And so why is that? And what about those neuroadaptations? Well, partly those neuroadaptations are due to the strong increases in dopamine that occur in response to drugs of abuse. And because of that dopamine, it's thought that the drugs of abuse hijack the normal reward learning mechanisms. And so that's another reason for looking at these synaptic mechanisms. All right, well, to begin with, we're going to be evaluating synaptic plasticity in response to two different temporal patterns. Now, a theta burst protocol was developed in my lab. This is 10 bursts of 10 trains of 40 stimuli, and those 40 stimuli are given as 10 bursts of 4 pulses. And if we use a theta frequency of 10 hertz, not 5 hertz, which is more typical for hippocampus, but that 10 hertz, which is the predominant frequency observed dorsomedially, then what we see here is a long-term potentiation, much stronger medially than laterally. Alternatively, we can give a 20 hertz stimulation, and that produces long-term depression. So we're going to investigate those molecular mechanisms, and we're going to model them. Well, what mechanisms should we be modeling? What signaling pathways? Well, us and many others have done experiments showing what molecules are involved. So we have dopamine receptors, of course, shown here. So this is the baseline in gray in all these patterns, and these magenta responses show the effect of blocking that particular molecule. So we block the dopamine receptors, we do not see plasticity. Dopamine leads to activation of pKa. If we block pKa, we see a reduction in the plasticity. Metabotropic glutamate receptors, if we block those, we see no plasticity. Metabotropic glutamate receptors lead to activation of protein kinase C. We block it, we see no plasticity. And of course, NMDA receptors are involved. Now, this and many other molecules are involved. So that makes it really confusing. Which of these molecules are important, or are a combination of them, or is it a sequence? And so that's what we can address with the modeling. Now, one of the issues is that we have diverse temporal and spatial scales. So this is showing you a small piece of dendrite with 2 spines, and we know that glutamate receptors are predominantly on the spines and lead to influx of calcium in the spine. And this calcium is mostly confined to the spine. And because of the size of these spines, there's very few molecules. So a 1 micromolar concentration of calcium would only be 60 molecules. And so that means that these reactions in diffusion occur stochastically. But many of the G protein coupled receptors are not just on spines, but also on the dendrite. And so producing, say, dopamine receptors activating cyclases to produce cyclic AMP, which can diffuse. And so we have to model larger amounts of space, not just single compartments and diffusion. And though there are only a few molecules in the spine, there can be many molecules in the system. So the small numbers of molecules in spines means we shouldn't really be modeling these things deterministically, i.e. using differential equations. And in addition, the diffusion means we need to have multiple compartments, and that is computationally intensive. So we've designed some software called NeuroRD. Just very briefly, we've implemented what I call a tau leap for diffusion. The tau leap for reactions was developed by Gillespie, who also developed the exact stochastic simulation algorithm, which is a very common simulation algorithm to use. So we implemented this tau diffuse. And then we also further developed the algorithm to dynamically switch between using the exact algorithm and this approximate and much faster tau leap. And so we ended up getting dramatic increases in the simulation speed. So I'm going to present simulations mostly using a small piece of dendrite with 1 or 2 attached spines, but some simulations to look at spatial specificity and spine interactions, which has a much longer piece of dendrite with multiple spines. And I'm going to use various types of synaptic input, of course, glutamate, where the calcium influx represents NMDA receptors, and of course the dopamine. So we started with just calcium and dopamine activated pathways. And the question was, can calcium and dopamine synergistically activate? And we focused on protein kinase A and CamKinase 2, which I've showed you the protein kinase A was required. And of course, CamKinase is a well-known molecule involved in memory storage. And so here you can see the calcium, which activates calcium, binds to calcium calmodulin and activates CamKinase. And then we also have this dopamine input, which produces cyclic AMP, which leads to pKa. And then we have a few points of interaction here. So we simulated the response to dopamine alone, calcium alone, and the combination. And this is the morphology we used. And what you can see is that with dopamine, we got a very strong protein kinase A activity. With calcium alone, we got a very strong CamKinase activity and very little pKa. And with both, we got both stimuli, but you can see there's really no synergy. So unfortunately, this question we tried to address was, we're not sure why you need dopamine plus glutamate. The fact that there's no synergy means you could double your dopamine stimulation and get the same amount of both molecules. Of course, it's possible and highly likely that these molecules are activating different pathways, both of which are needed, but this model could not address that. Well, we wanted to know whether these kinases could discriminate temporal patterns. Remember, I told you that with theta bursts, we get LTP, but with 20 hertz, we get long-term depression. So we'd expect to see a difference in these kinases. So CamKinase does exhibit a small difference between theta burst and 20 hertz, but protein kinase A does not. And if we look at AMPA receptors, which is one of the downstream targets, and there are two different phosphorylation sites, we only see small differences. Okay, so we said, all right, we have to look at other pathways that we know are involved. And in particular, there's a set of GQ coupled pathways. Okay, so metabotropic glutamate receptors, muscarinic M1 receptors, activate this G protein of subtype Q, which leads to the activation of protein kinase C, and also 2AG. 2AG is an endocannabinoid, and numerous experiments have shown that this molecule is required for long-term depression. So we need to look at these. So now what I'm showing you here is 2AG in response to theta burst in 20 hertz and protein kinase C. And you can see a dramatic difference. The 2AG is sort of similar. The pattern, temporal pattern is a little different, but overall quantity is similar. But the protein kinase C is much greater with the theta burst than with the 20 hertz stimulation. And we can summarize that here. This is showing you total or the mean concentration of 2AG with theta bursts and 20 hertz. They're a bit similar, but with protein kinase C, we have a much larger PKC quantity. And I showed you that PKC was required, but in fact, we did this experiment after we did the model and showed that indeed, if you block PKC, you block LTP. Now we use this longer dendrite with a bunch of spines to try and investigate whether we had spatial specificity, because one of the key tenets in synaptic plasticity is that only those spines that are stimulated should undergo plasticity. So what I'm showing you here is, again, the morphology, and then the y-axis is the spine, top and bottom. The top shows you theta burst, the bottom 20 hertz, and then this is the time. And so the darkness of the graph indicates the concentration of 2AG. And you can see for theta burst, you can see that with each burst, you see a little bit of an increase in this 2AG, but it's not just limited to the stimulated spines. there's sort of this gradient, this small gradient, and it's spine 1, which is stimulated in this case. And 20 hertz also has no gradient. This suggests that when you induce LTD, nearby spines could also be depressed. And this might be a mechanism of compensatory heterosynaptic plasticity. But PKC has very high spatial specificity. You can see for theta burst that mostly the stimulated spine has high concentrations of PKC. But for 20 hertz, we don't see much at all, but we still see this gradient. So then we wanted to merge these pathways. So these are the GQ pathways that I showed you previously. And here are the GS pathways that I showed previously. So again, we have calcium coming in, we have dopamine, and we also have inputs of acetylcholine, which acts on both. It inhibits the cyclic AMP through this pathway, and also we have our GQ pathways. And I didn't show you this before, but in all cases, we constrain our model to experimental data. So there's been lots of measurements of DARP32 phosphorylation on either threonine 34 or threonine 75 in response to dopamine or calcium or the combination. And so what I'm showing you here is our model response. And this is very similar to the experimental responses. And then for the GQ coupled pathways, we have some experiments measuring the effect of how much depolarization will suppress inhibitory and excitatory inputs as a function of activating metabotropic glutamate receptors and also a stimulation time. And then this is our model. We're measuring 2AG because this experimental phenomenon is dependent on 2AG. What about our inputs? Our inputs are also constrained. So Armando Salinas in my lab, in collaboration with David Levenger, measured whether dopamine produced a higher response with a 20 hertz stimulation versus theta burst. There was not that much of a difference, but other published data shows that dopamine decreases if you give trains of stimuli or pulses of stimuli very close together. And this shows that the longer you wait, the higher the dopamine on the second pulse. And so we use this data to constrain our dopamine signal inputs. Now what we're showing you with this combined model is phospho-cam kinase up here in the spine. And you can see this very noisy data is because of the stochastic fluctuations and in the dendrite. And in this combined model, we see a much larger increase between theta bursts and 20 hertz. We also see a similar PKC response in the spine and again in the dendrite, much greater for theta bursts than 20 hertz. We also evaluated protein kinase A and another cyclic AMP activated molecule called Epac and the 2AG and the dendrite. Now, once again, we're left with, how do we put all these molecules together? Is only one of these molecules? Do we only look at the CAM kinase? Do we also look at the 2AG? What's the role of protein kinase A and Epac? And so what we did was we used just some statistical analyses to try and develop a combination of these key molecules that would best predict not just the plasticity for theta burst and 20 hertz, but also experiments where different blockers of molecules have been used. So what I'm showing you here is 3 trials for each experiment. So often you'll see the clusters of these symbols and different theta burst protocols that produced LTP with or without different blockers, various theta burst protocols that had no plasticity. Again, this was produced by various blockers, and then 20 hertz that produced LTD. And I'm plotting the total PKC in the spine versus phospho-cam kinase in the dendrite, or PKA phosphorylation in the spine versus 2AG in the dendrite, because the model said that these are some key molecules involved that could predict the outcome. And so what you can see is that when protein kinase C and CAM kinase are high, we're predicting LTP. And when CAM kinase is low, we're predicting LTD. And on this graph, we can see that low 2AG leads to LTP. Now I want to ask the question about what happens with ethanol use. So it turns out that alcohol withdrawal, and even long after, such as a month after withdrawal, and most of this work was done in rodent models of addiction, there are persistent changes such as enhancements in NMDA receptors, changes in those GS-coupled pathways. So we implemented in our model these molecular changes. And then we didn't do the full protocol. We said, well, in relapse, remember I told you that a single trial can reinstate the alcohol use. So instead of giving the full protocol, we gave a partial protocol. And under control conditions, a partial protocol of theta burst will produce LTD, and of 20 hertz will also produce LTD. So we don't see LTP. And then we've resimulated this in the withdrawn condition. And these arrows are showing you the change in the signature. And then the fact that we have these solid symbols means that we change was the new outcome. So in response to a partial train of theta burst in our withdrawn model, we now get very strong LTP. We have higher protein kinase C, higher CAM kinase, higher protein kinase A. With 20 hertz, we also see a change. In fact, what we're seeing is with 20 hertz, instead of LTD, we are now seeing LTP. Now, these are consistent with experiments that show that LTD is not produced with 100 hertz stimulation, which is another type of LTD protocol in the striatum. But it is a true prediction because these experiments have not yet been run, but we're hoping that my collaborator will do those. We wanted to revisit the question of spatial specificity. So again, we have this long dendrite with several spines, and I also want to look at whether these spines interact. So spines 0 and 1 right here, or we can stimulate spine 0 and 6. So this would be the adjacent condition, and 0 and 6 would be the separated condition. So what we're looking at here, again, is some of those molecules in the signature, protein kinase C and protein kinase A. The darker traces show the stimulated spines, and the lighter traces of the non-stimulated spine. So you can see that the molecules go up much greater in the stimulated spine than in the non-stimulated spine. And this is for theta burst. So this is consistent with our prior observations where we see spatial specificity for long-term potentiation. But you can see that we don't see much spatial specificity for 20 hertz. Now, the black versus red is the adjacent versus the separated spines. And what we see is, at least at this spatial scale, we're not really seeing much of an adjacency advantage. Now, remember, we don't have the electrical activity. So there still could be an adjacency advantage if our calcium influx was changed based on some electrical cooperativity. So we can't rule that out. When we look at the dendrite, we don't really see much spatial specificity. So again, the top is the adjacent condition, and I'm pointing out the stimulated spines. Again, left is theta burst, right is 20 hertz, and then the bottom is the separated spines. And the fact that you can't really pick out which spines were stimulated is why there is not much spatial specificity. So in summary, I showed you that several molecules, CAM kinase, Epac, pKa, and protein kinase C, are elevated for theta burst compared to 20 hertz. And a combination of these molecules can predict the response to various stimulation protocols and inhibitor treatments. I also showed you that withdrawal from chronic ethanol, which we tested with weaker stimulation protocols, also showed that we're going to get enhanced LTP and we're going to have a altered response to 20 hertz. Namely, we get LTP instead of LTD. And so this change in signaling pathways and change in plasticity can contribute to the relapse in response to single cues. And you could say, well, how do we make sense of all these molecules? I just showed you a linear model. Well, what we've been doing in with our hippocampal signaling pathways, but which is a future direction in the striatal signaling pathway, is look at some of the key downstream molecules that integrate all these kinases. And one of those is ERK, which is an integrator of plasticity. And I hope to show you in a few years that if we can bypass looking at all of these molecules and really just focus on a few key molecules such as ERK to make predictions of whether plasticity will occur. And I have to end with acknowledgments of the people who did all this research. Spatial, who wrote the NeuroRD algorithm, Sarah Hawes, who did the experiments, Rodrigo, who did the calcium and dopamine model, Blackwell Kim and Lane Wallace, who did the GQ coupled pathways, and the rest of these people who contributed in other ways. And of course, my collaborators, Jeanette Helgren-Kotaleski and David Leppinger.